What drives me is moving science forward, trying to figure out how to get an answer to a question in a new way, try to work with a sample type that we didn't necessarily think that we could, try to move an experiment forward that people didn't necessarily think were possible. My name is Marie Adams. I am the Genomics Core Director at the Andel Institute, which is a private research institute in Western Michigan. I am essentially responsible for coordinating all of the activities that happen with regard to whole genome sequencing, RNA sequencing. One of the biggest challenges is that we have to be really nimble. We have to be a little bit of everything for all people, and that is sometimes really difficult to do when you're trying to also have really rigorous and reproducible assays. One of the experiments that we were really interested in looking at was a co-expression of human and microbial material out of the same RNA sample. There aren't necessarily a lot of kits on the market that make it possible to do this in a very cost-effective manner, because we need to do the ribosomal reduction of not just the human sample, but also the microbial sample in order to look at the underlying messenger RNAs that are present. Without the ability to do that ribosomal reduction, we were basically wasting a lot of sequencing reads, wasting a lot of sequencing depth, and slowing down the experiment from getting back to the researchers so that they could do the analysis and make their scientific decisions. The QIAseq FastSelect allows us to remove the intervening bacterial ribosomal sequences so that we can actually focus on the underlying mRNA that's there in the sample. It also is a really neat drop-in solution into our already existing RNA-seq workflows. So we didn't have to revalidate our experiment, we didn't have to revalidate our assay. We just could basically pipette it in, do what we needed to do, and call it a day. It was a really fast solution. Usually ribosomal reduction adds a significant amount of time to the library prep procedure, but since this isn't an extra step, it's part of the same step that we already do in a different part of the process. It allows us to keep our turnaround time as fast as it usually is. We don't have any delays. My biggest wish would be to have the human mouse rat and the bacterial ribosomal kits to be put together in sort of an epidemiology kit. The other thing that we would really like to see, obviously, is an expansion of the things that are available in the panels themselves to not just human mouse rat and bacteria, but to more diverse organisms.
QIAGen.
Sample to insight.